thank you guys for having me and for the invitation. Yeah, I'm kind of excited to present my talk and in the context of the last couple of talks. I think we've heard a few instances of how context really matters in biomolecular folding and function. We've heard about gravity, eutectics. This one, kind of framed it as Mars, but really I think broadly it's a story about salt and some of the impacts that we've seen that it can have that are really unexpected on how we saw biomolecules function, particularly nucleic acids, but we've seen some effects on protein as well. And so, yeah, I think there's synthetic biology implications of the talk, but this is kind of the genesis of the story and where we were living when we were thinking about the problems that we were trying to solve here. So this is a really nice diagram from Jerry Joyce from about 20 years ago, showing kind of when things were thought to have happened in the emergence of life on Earth. You can follow it, and that time scale is billions of years ago through the formation of Earth to having a stable hydrosphere. And then that underscored area is kind of a lot of what we think about in terms of prebiotic chemistry, where small molecules can come together, assemble into things that start to look lifelike, things that you might have as components of an early cell. Going through some kind of RNA world, I think there's really been a shift in the last 10 or 15 years in terms of where proteins might have come in there. I certainly would allow some proteins into that 4.0 to 3.8 billion years ago area. I don't think it's necessarily pure RNA, but I think we're certainly looking at a time where RNA did a lot of work that in the past that it's largely been supplanted by proteins now. So this is kind of the thinking of how it looks on Earth. We've been looking at Mars, and this was kind of motivated by some discoveries in the last 15 years ago or so. This kind of came about from some work from the Phoenix mission. It was later validated by several other Mars missions, in which they found that there is a lot of an ion called perchlorate on the surface of Mars, in the regolith, or dirt, if you like, on the surface. And so this came about with this Phoenix mission, where they originally went looking for nitrate. They did a series of experiments where basically what they went up with showed that if they were trying to explain the result that they got by nitrate, there would actually be more nitrate than there was mass of regolith, and they were trying to analyze. And it turns out, based on some ancillary experiments that they did, that there is a lot of oxychlorine salts on the surface of Mars, chief among those probably being perchlorate. And this is really interesting from the perspective of how you might get life on Mars, how life might sustain itself on Mars. One key element to this being the fact that perchlorate really likes to hang on to water. If anyone here is a bench scientist, they've probably seen this phenomenon. So this is just looking at sodium hydroxide pellets. This is what happens if you're in the northern hemisphere and you're thinking about weighing these out during the summer. If you weigh them out, you get these nice little prills that you have on the left there that are kind of dry. They start to look shiny if you don't work quickly. And if you leave them out for a long time and don't dissolve them when you're planning to dissolve them in, you end up with kind of a puddle there. And so this phenomenon is called deliquescence. That just means that there's salts that or compounds that really like to hang on to water. They pick it up out of the air. This is something that we see in the lab. There's also some evidence that this is a process that might be happening even on present-day Mars. We know that perchlorate salts are incredibly water-loving. They can do this process where they pick up water from the air. There's been some evidence presented that there's actually cyclic processes causing this on present-day Mars, where there might be brines transiently forming and basically wet-dry cycles that are happening. And so, if we think about life on Mars, if there's some kind of life that's been continuously sustained from a past wet Mars, or if we're thinking about life emerging right now, there's probably these brines around, and life has to get by with these and sort of function with them. If we look at examples of extremophiles on Earth, there's a number of them that basically pump out salt in their cytosol to kind of have a more mesophilic-like environment. There's also a class that when they have to get along with a ton of salt, basically live with having a really salty cytosol. So what we were thinking was this would be a really powerful selection pressure for molecular evolution on Mars, whether that was an early event, this is something that's been around for the whole time if there's extant life on Mars, or if this is something that is later that life has kind of evolved along with it. Biomolecules presumably would have to live with a lot of these salts. And so we looked at sort of how some of these prebiotic type processes would get along with this level of salt. And so if you look at perchlorate, you can rank it on this, what's called the Hofmeister series. And so this is a series that was developed by this biochemist, Franz Hofmeister, that basically characterized a series of salts and showed that they have a lot of different effects on biomolecule folding, solubility. If you've done protein purification, they have something to do with that. We were principally interested in it for the impact on stability. If you look at how biomolecules fold in the presence of these salts, this is a plaque commemorating this Hofmeister series. It's actually displayed in Prague, showing you can follow this from organic ions down to chlorate. Perchlorate would be among the most destabilizing of those. If you look at biomolecules in the presence of some of these salts, you can see that they kind of follow this Hofmeister series. So this is the melting temperature of lysozyme, kind of a typical model protein, in the presence of varying concentrations of some of these Hofmeister ions. And so sulfate and phosphate would be on the left side of that, the more stabilizing ones. You can see that as you add it in, the melting temperature goes up. Chloride is kind of around the neutrals area where you don't see much change at all. And then iodide and perchlorate are towards that destabilizing side where you can see that the melting temperature goes down in a pretty pronounced fashion as you add in more perchlorate. This is only going up to about 1 molar. When we're talking about these brines on Mars, we're contemplating multiple molar concentrations between 3 and 10 molar, depending on the salt. And so what we did was kind of inspired by what might be happening in some of these salt-adapted organisms, looked at exactly what might be happening with biomolecules in the presence of perchlorate. One thing that can happen with them is these salts can really drive adsorption to solid surfaces. If you've ever done a plasma image prep, this is actually using some chaotropic salts that drive adsorption to silica. This is basically how this purification process works. This works with essentially any silica-based resin, so pure silica, diatomaceous earth or pumice, or even people have done it with pulverized light bulbs. Basically, just glass will do it. We've done this with a simulant that looks at. This is actually what I'm showing on the screen right now. So this is the Mars Global Simulant. So this is basically an Earth-based compound that uses some basaltic minerals on Earth that give sort of the best general purpose analog of simulant in one of these sites on Mars, the Rocknest site, where we've characterized kind of the mineralogy. What we found is that you can actually do sort of this plasmid purification type process with perchlorate. What this gel is basically showing is essentially that we can get perchlorate to, or using perchlorate, we can get nucleic acid to stick to this simulant, or we can get it to stick to silica and then reversibly elute it. And so this is a process by which you can see that nucleic acids will actually kind of reversibly adsorb to it. And this is one process we can see happen that's sort of promoted by these brines. So another thing that we've looked at is basically how nucleic acids can fold in these, looking at how an RNA world might be influenced by some of these really salty solutions. This is just looking at a double helix of DNA, and you can see that as you increase perchlorate, as you go from low to kind of moderate concentrations up to about 1 molar, you get an effect that's sort of predominated by salt effects. And then as you go to really high concentrations, you can see that you get a really pronounced decrease in the melting temperature. So you can see that we see kind of this affected destabilization effect associated with perchlorate. We go from something that had, a 70 to 80 degree TM all the way down to, roughly a room temperature melting temperature. This isn't true of every single secondary structure. What we found is that if we look at some non-canonical secondary structures, we see some really different effects. The red structure there, also shown with the red line, is a 4-stranded G quadruplex structure. So this is one of these non-canonical structures that's found in promoters for a lot of proto-oncogenes, telomeres in eukaryotic genomes. And what you can see is that this is much less pronounced. It looks like there's some kind of privileged structures that exist uniquely well in these sort of high salt chaotropic solutions. We can actually use this to sort of modulate the folding state of nucleic acids. So this is basically looking at a set of 2 strands of nucleic acids where we have a G-rich one that can fold up into this 4-stranded structure, as well as its C-rich complement. And so we've got a situation where you can have that state A, which is the C-rich and the G-rich strand forming a double helix. A second state, this B state, where the G quadruplex folds on its own, so we can have single-stranded folding. And then a third state where the two strands come apart and we've got the single strands there. And so this is basically using a fluorescence monitoring experiment, a FRED experiment. We can follow these depending on salt concentration and temperature. And what you can see is if we interrogate these at different salt concentrations, we can kind of pick, depending on temperature and salt, which one we end up in. You can see that if you're at the lowest temperature in low to medium salt, you're in that first state A, the duplex state. Or at the high salt concentration, you can see you're in that G quadruplex state. That one is only going to go from that G quadruplex state to an unfolded state. And then the other ones can actually sample in the middle there, that G quadruplex state. And so we can actually have strands of nucleic acids that, depending on salt and temperature, will go through multiple folding states. We can also catalyze some electron transfer chemistry with these. It's known that these four-stranded structures can actually bind to heme, just like a protein peroxidase, and catalyze single electron transfer reactions. We can use dyes to interrogate these. And what we found is that even in these really salty brines, you know, at about 5 molar, what you're looking at there is as that A570 value goes up, that's generating the dye that indicates electron transfer is happening. We can perform electron transfer chemistry. And so even in these really salty solutions, this is chemistry that's possible here. So, you know, even though we think of these as being fairly denaturing, we can perform, you know, some chemistry that is involved in a lot of biological processes. Another thing that's interesting about this is that we found that the perchlorate actually isn't degrading molecules you would expect to be redox active. We could put an antioxidant in there that's very sensitive to the presence of oxidizing species. If we put hydrogen peroxide in there, you can see that it's pretty much consumed over the course of a day. Perchlorate, despite, you know, kind of having this reputation as a strong oxidizer, is kind of metastable, and it actually won't undergo reaction at least a significant rate. We can have it around for several days with these antioxidant species. And so, you know, despite being something that has a really high oxidation potential, on the timescales that we're looking at it, it's not actually reacting with biomolecules. Another thing we've done with some of the electron transfer chemistry is found that we can do some chemistry that actually hasn't been done with ribozymes before. So, you know, I mentioned perchlorate was one of the oxychlorine species that's thought to be present. Chlorate is another one of those. What we found is that if we take chlorate, that can act as an electron acceptor, but can also act as a chlorine donor. And if we use that same G quadruplex heme complex that we showed could do electron transfer reactions, we can actually do chlorine transfer reactions with this. And so, to our knowledge, this is the first case that someone's seen a carbon-chlorine bond formation with a ribozyme. And this is actually using one of these Hofmeister sort of Mars-relevant ions here. So we're really excited about this, both from the perspective of Mars, as well as the fact that you know, this seems like something that could potentially really extend the suite of reactions that's possible with ribozymes. You know, if you look at the tremendous success of ribozyme chemistry that we've seen over the last 30 or 40 years, despite that, a lot of the reactions that we know ribozymes do tend to be phosphate transfer reactions. We know of some cases of electron transfer reactions and some carbon bond forming reactions, like the Diels-Alder reaction, but it's not nearly the same suite that we enjoy with protein enzymes. And this is an example that we can show kind of extends the suite of chemistry that's sort of possible with ribozymes. We can also use some other functional ribozymes in perchlorate. One thing that we found is that, you know, again, despite being something that's nominally denaturing, we can take the hammerhead ribozymes, so sort of one of our prototypical functional RNAs. It's a nuclease. And so that's those two strands together will cleave at that red arrow. We can follow the rate there. So that line, the more it goes down, the more it's working. And you can see that in both chloride and perchlorate solution, it actually retains function up to, you know, molar concentrations. We can use perchlorate to regulate function as well. One thing that we've seen is that, you know, we have this ribozyme that works, but this might not be something that you would always want to have switched on in an origin life reaction. One thing that we've done previously is shown that if you have liposomes that are growing in the presence of really short oligonucleotides, those oligonucleotides can inhibit a ribozyme's function at high concentration, but as a liposome grows, it can switch it on basically as the dilution associated with the lumina liposome growing decreases the local concentration and allows the ribozyme to assemble because those inhibitor oligos dissociate. We actually see that this can happen with perchlorate too. If we have a lot of those inhibitor oligos around, we'll see that the ribozyme switched off, but then as we add in perchlorate, we can actually switch it on. And so this is something where, again, we could see something where we have kind of conditional function or regulation in one of these that happens at as you go from essentially high to low concentration. So something that you might see promoted by a wet-dry cycle. We can use more complex ribozymes in these perchlorate solutions as well. This is ribozyme polymerase, the TC19Z ribozyme polymerase. And what we found is that, it works up to, a few molar in sodium chloride. We can go to almost that concentration in perchlorate and basically read through and copy a decent length RNA. Another thing that we found is that this actually will enable us to read through some secondary structures that have proven problematic in ribozyme copying. So this is basically challenging that polymerase with reading through a hairpin. That's something that these really struggle with because relative to protein enzymes, they don't have things like helicase domains. And so we speculated that it might be the case that this sort of modest denaturing function of perchlorate might allow for read-through of these. And this is something that we see. You can see relative to chloride, which again is that one that's kind of neutral on that Hofmeister series, we can see read-through of a secondary structure that basically shuts down copying. So this is another case where these ions can actually enable new functions. We can see this with some other nucleic acids. A fluorescent aptamer will also retain function in perchlorate up to several molar concentration. So this seems to be a general phenomenon with a lot of these ribozymes, even though that they were selected in kind of low salt conditions. And when we compare protein enzymes, this is really not the case. If we look at EcoR1, so this is the top one, a restriction enzyme that works in a mesophilic chassis, it shuts down around a couple hundred millimolar perchlorate. And it's only when we look at things that were selected in halophiles. So this bottom one is a beta-lactamase that was selected in there, that evolved in a halophile. And that one will hang on up to several molar perchlorate. So really only competing with the nucleic acids when we look at the ones that are really highly evolved for high salt. Same for renaturation. If we go to sort of prohibitive concentration of salt, we can shut proteins and nucleic acids down. The nucleic acids and the halophilic or extremophilic proteins will recover function, but not some of these mesophilic proteins. So again, it looks like RNA is kind of uniquely well adapted to some of these high salt solutions. So that's kind of what we've been doing with some of these. We've seen some cases where, it looks like nucleic acid function is, certainly not prohibited in some of these high salt solutions. And additionally, we've seen some cases where some new functions really switch on. We can see the regulatory behaviors work. We can see nucleic acids work in cases where proteins sort of struggle, or they only really work in the case where they were evolved in extremely high salt. And then we can see this chlorination reaction that, again, we're really excited about, not just from the perspective of perchlorate kind of high salt solutions, but in terms of expanding the chemistry that nucleic acids can do. It looks like this is a function that was sort of latent and not present, or not known to be present, and I think could enable a lot of really interesting chemistry. If you think about chlorination reactions, that's sort of a foundational reaction in synthetic organic chemistry, and I think that this has some really strong implications for things that we could do with nucleic acids. All right, so thank you guys for your attention. Tanner really spearheaded a lot of this work along with Matt, Ben, and Nate. It's a collaboration with Kate as well. Funding through the Center for the Origin of Life at NASA, Heising Science Foundation, and Research Corporation for Scientific Advancement. And you guys as well for your attention. Thank you again.